Good morning. It's a little bit awkward because I think that, like, I don't know, maybe people are a little bit anxious. I know that for those of you that are not following along with the news, so we had a transmissive incident here where we have had a small outbreak. And obviously we don't know how many people are involved in that yet because we only have those are the only people that have tested. So I don't know where in the school it is. Maybe you guys have more information about that than I do. People talk. But anyway, well, I don't know. So nobody's told me. But anyway, it's a little bit concerning because my assumption is that people are already following the health and safety protocols, and the transmission here was between a student and a teacher, or from teacher to student. I don't know which way it went, but so that's obviously concerning. That people probably have a little bit of anxiety around that. I don't know if there's going to be any change to the how we're operating on the ground, if there's going to be a change to school policies or what we're doing. There's supposed to be a staff meeting at 10 o'clock today, so I'll get the notes from whoever is going to attend. That I can't obviously go because I'm going to be here, but there'll be a discussion about that. I think maybe for today we'll put our school walkabout on hold. I don't know if that's supposed to change or not, so maybe we'll just skip it for a day. And then we'll reevaluate that one tomorrow. But other than that, I think everybody in here is doing what we're supposed to be doing. Let's keep doing that. I have fairly high confidence in our health and safety protocols and what we're doing here. So, and so does my partner, my physician. So. I think let's just keep going. There's nothing really else to be done here. So, but if you guys do have questions or if you're concerned about something in particular, we can definitely talk about that too. So feel free to ask if you feel like you have a question about something related to that. Okay, let me do attendance real quick and then we'll get started. How far did we get? We were doing that survey of genetic traits, right?
Yeah, we were
Okay. Cool. We'll pick that up in 2 seconds. So if you want to throw that up on your screen, we're going to get to that in just a second here. I'm just going to take attendance quick. What's that? Oh, right on. And just remember, this is for the people at home. If you're not attending because you're concerned or whatever, that's fine. Just please make sure that your parents are calling the school to let them know why you're not at school. You can totally do that if you want, though. Okay, was it on Jaden? Listen. Okie doke. So some quick things to mention. Tomorrow is the last day of this unit. I know it feels weird. So we are going to have a little project that we complete in the second half. I noticed that there are a few people who haven't handed in the project from the first unit yet. That really has to be done, guys. I'm not going to chase you down on it, but it's got to be, and I already marked the other ones. So I should return the marks to the rest of you guys. Sorry, I'll send them back in a bit. But and actually at the end of this block, give me a couple days and I'll give you a mark update too. I'll put it up on so you can actually see what your mark is in the course. I obviously don't have that much evaluative information, but I mean, like there's going to be less evaluation than normal. Otherwise, you guys would be working 24 hours a day. So. Where was I headed with this? Oh yeah, right. Which means that we're going to have a unit quiz tomorrow. So it'll be just like the last one. It'll be, I think it's 30 multiple choice. I got to double check, but it'll be in Learning Block 3. You can just write it at home. It's open book. Okay, just like the last one. And the vocabulary list, hopefully you're on your way to creating a vocabulary list for this unit as well. I know this stuff is coming really fast. It's just like, oh, I thought we just handed in the last one. It's like, yeah, that's how the course works now. And it sounds like a few days later and we're done another unit. So that's going to come due in the next day or two. And then that project will be due at the end of Thursday, which I guess is technically like a day out of your cycle here. But if you could finish it earlier, that'd be ideal too. So that means Wednesday, we're going to be starting the next unit, which is animal systems. So we'll be well on our way. One day of animal systems, one of these days in animal systems is like well into the unit. We'll be probably finished the digestive system on Wednesday. So anyway, if you're wondering where we're headed here, let's, and then you get a break. So I don't know where you're going for your other course. Maybe it's more work. I don't know, math or I don't know where you're headed, but okay. Next up, what did we just do? We just did thumb straight, right? Oh, I forgot to do my little calculation on here. So what percentage of the class was thumb straight? 5 of 16. 31.3%. Cool. So about a third, a little less than a third. Okay, bent little finger or straight little finger. So in order to figure this out, you got to hold your hands so that the palms are facing you, like this. And then if your little fingers bend inward, then you call that bent little finger. And if your little fingers are like straight butted up against each other, like you've got straight right there. That's a perfect example of straight. Mine's a little hard to see, but I have bent. So there's like a little V at the top of mine when I push them together. So if you've got a little V at the top, you're bent. And if they're like touching each other at the top, that's straight.
Mine's like got a little curve.
Yours is straight. Yours is definitely straight. When you're pushing them together, there's no little V at the top between your two pinkies. Okay, like this. So you'll see like a little V. It's not this pronounced. Like they've exaggerated it on this diagram here. But if you have a little V at the top, that's, what are they calling it? Curved? Bent. Okay, this is a genetic trait too. It probably has something to do with bone or ligament formation or something. I'm not sure the actual genetics behind this one, but okay. So bent in this case is dominant, and straight is recessive. So I have bent finger. That means that so that's my phenotype. My phenotype is what it physically shows. I'm going to try to use this terminology as much as possible so we really get into it here. So that's my phenotype. My genotype, or my set of genes that gives me this, or my set of alleles, is big B, little b, or big B, big B. I don't know. I have the dominant gene, but I don't know what the second one is. It could be another dominant or it could be the recessive, but both of those would show dominant. So I have bent. If you have the dominant, that is, you have bent little finger, throw a hand up for me. Sorry, people at home. Too hard to count you. 1, 2, 3, 4, 5, 6. Oh, okay, that's a fair amount. I got 10 here. Now we have fewer people in the room this time, so that kind of throws it off. I got 14 now. Okay, so that's 71.5%, 71.4%. So just because something is very common in the population, as I mentioned, like these traits are less, the dominant traits less and less common as you go down through this list. But that doesn't mean in every population that you survey it's going to be exactly the same. Obviously, there's like a fairly random distribution of this in the population, so you never know how you're going to sample. Technically, if we were to sample like a really big crowd, like of 1,000 or 2,000, I think you'd start to see the stats of that bear out, that there's progressively less and less. Certainly the first one, a lot of people have that dominant trait. Okay, widow's peak. So widow's peak is when your hairline comes to like a, in the front, a little, I don't know what to call that, a peak. Does that make sense? If you have a little peak in the front. So this one you kind of got to like pull your hairline back, have a look, as if you've never taken a selfie before. But you know if you got it up there, right? Maybe you don't. You pull your phone out and take a look. I definitely have a widow's peak. It's more pronounced on some people than others. If you're not sure what I'm talking about, let me find a really good actual picture of somebody with a widow's peak so you can see it. Oh, there she go. Let's just make sure my stream is working again. Can you guys see this again? It looks like it's streaming. Just type something in the chat. I'll wait till I hear from you guys. Oh, it's a real pain when it gets disconnected. You can see it? Yeah. Okay. I'm just going to restart it because the chat stops working. Okay, there we go. Oh, what did I just do? 1 sec, guys. When it works, but when it doesn't work, it really doesn't work. Okay, there we go. Widow's peak. Yeah, there's a good one. That. So does it come to a little point at the front? Whoa, what a picture. Oh, there you go. Leo's got a real good one. All right, let's survey. I have one. Who's got one? 2, 3, 4, 5. 5. All right, not that many. Okay, it's 35.7%. So my phenotype is that I have a widow's peak, but because of that phenotype, I don't 100% know my genotype, but I have at least one dominant allele. So I definitely have at least one allele that says widow's peak. Okay, dimple chin. Dimple chin is when you got like that little imprint in your chin. Is that what I'm talking about here? Often when we do this, people don't know what I'm talking about. Oh my goodness, there's a real serious dimple. Most people, okay, that's like an extreme example. Here, this is a good one. This guy's got it. So if you got like, oh, there's another good dimple. Boom. Little imprint in the chin. Okay, so that's the dominant trait. So I don't have the dominant trait. That's my phenotype. Big D is dominant, little d is recessive. So because I don't have it, my phenotype is no dimple. My genotype, I know for sure, is both alleles are recessive. What do you have? Do you have a dimple? Question mark? Put up your hand if you have one. No one has one in here. There's usually at least one. Really? Not a single one. 0, 14. That's easy to calculate. Huh. Usually there's at least one. Okay. Mid-digital hair. This is a little bit of a weird one. So if you look at your fingers, there's like 3 sections to your phalanges, to your fingers. You got your first section, and then you got a middle section, and then you got the fingertip. The middle section, have a look at it. Does it have hair on it? This is for boys and girls. Doesn't actually matter. And they could just be little stubble hairs. They don't have to be, I mean, obviously you're not gonna have great big hairs growing on your fingers, but.
What if it's only one finger?
Is there hair growing on it?
There's a little bit.
You got it, yeah. That doesn't have to be a lot. I've never heard of anyone having it on only one finger, but it is possible. So mid-digital hair is the dominant trait. That's a capital H. And no mid-digital hair is a little h. I definitely do have it. All right, who's got mid-digital hair in here? If you don't mind saying. Not that it's weird or anything. Lots of people have it. 1, 2, 3. Just 3. Connor, is that a hand up? Mm-hmm. It is. Okay, 4.
Let's go, boys.
So 4. I have it as well, so that's 5. So that's 36%. Have the mid-digital hair. Is it all guys? Oh, that's interesting. Usually a lot of girls have it too. Huh. I didn't count a girl. Weird. Okay, and last one. So this one is interesting because eye color is a polygenic trait. Okay, so like what is your eye color going to be? That's not determined by one single gene. So all these things that we're talking about here are Mendelian traits. A Mendelian trait means one gene that you have is going to dictate whether you have it or don't have it or like what it looks like. Okay, and eye color is one of those polygenic traits. It's not just one gene, it's a whole bunch of different genes on a whole bunch of different chromosomes that tell you what your eye color is going to be. But one aspect of eye color is Mendelian. That is, one gene dictates it. And that's whether or not you have light eyes or dark eyes. Okay? So basically what it means is, do you have a lot of pigment in your eye or do you have very little pigment to no pigment in your eye? Okay? So if you have blue eyes or blue-green eyes, that's virtually no pigment in your eye. Okay? Or sometimes gray, like really light gray. If you have brown or hazel or any eyes that are not light colored, okay, it usually means that you have a lot of pigment in your eye, and that difference is dictated by one single gene, really light or dark. Yes?
Does it affect our vision though?
It's interesting. It actually does have an effect on vision. So humans probably didn't have lighter eye colors until fairly recently in human evolution. It doesn't have a lot of impact on you, but what it does impact is how well you can control how much light gets into your eye. Dark pigments absorb more light. So in very bright light, you can actually see better if you have darker colored eyes because you're better at blocking the excess sunlight from getting into your eyes. So people with blue eyes don't do as well at seeing things in extremely bright light. So that's probably a more recent genetic trait. It hasn't been essential for us as humans probably since the dawn of civilization to have like excessively good bright light vision. But if you're like living in a hunter-gatherer society, it's kind of more important that you can see really well in bright light. So that being said, it's possible that people with brighter eyes, I don't know this for certain, but it's possible people with lighter eyes have better vision at night because they could let more light in a dark environment. But I'd have to look into that. The converse is definitely true though. Okay, anyway, I have dark eyes, mine are brown. If you have dark eyes, that means you definitely have at least one gene for lots of eye pigment. They use P's. Again, P is a terrible letter to use for genotyping because it's really hard to tell the difference between a capital and a small letter P. So don't use letters like that if you can avoid it. This is not my naming scheme, but anyway, this is my phenotype, dark pigment. And so therefore my genotype is going to be one of these two, but I can't tell which because I know I have at least one dominant. Okay, do you have that? Who has dark colored eyes in here? 1, 2, 3, 4, 5, 6. 7 plus me. It's half the class. Interesting. All right. So the purpose of this is twofold. Oh, question?
What was the percentage for widow's peak?
Widow's peak was 35.7%. Yeah, so this is interesting because, well, for a couple of reasons, but what I want you to get out of this activity is a couple of things. One, humans have some Mendelian traits. There are traits where a single gene that you have is going to impact what your physiology looks like, your phenotype. And then the other thing that's interesting out of this is that, I shouldn't say interesting. The other thing that I want you to get out of this is in general how a dominant and a recessive trait works. So if you show the dominant trait, that just means that your genotype has at least one of the dominant alleles, okay? If you have the dominant, you're gonna have a dominant somewhere. If you're showing the recessive, that is you have the other one, like bent pinky, then for sure your genotype is only going to be recessive. So dominant always overshadows recessive. If you have the dominant gene, it overshadows the recessive gene. The recessive won't show if you have the dominant allele somewhere. And you have 2 copies of every gene, one from your mom and one from your dad. Okay, that's the whole point of the homologous chromosomes that we talked about earlier. There's 2 copies of everything. The only thing that you don't have 2 copies of, well, actually, girls, you have 2 copies of everything because your X chromosomes are homologous, right? You have 2 X chromosomes. But guys, you only have 1 X chromosome and 1 Y. So things on your X and Y are not homologous. They're only determined by one single allele, either on your X or on your Y. So you don't have a backup copy. That's going to play out later on when we talk about some traits that are related to being male or female. Color blindness is a good example of something that's found on the Y chromosome because you don't have a backup copy. So if you have a malfunctioning gene on your X chromosome as a guy, you don't have another one to potentially be functional. Girls have a backup copy. All right. So what you can do on the next page, you guys have this as well, right? Or is this at the bottom? This might be at the bottom. Is you can kind of like trace your path to see how common your particular combination of phenotypes is. So you start at the top and decide, okay, like do I have the dominant for tongue rolling? And it doesn't use all of them here, it just uses a few of them here. Or do you have the recessive, can't roll your tongue? And then from there, do you have the dominant or recessive of, what was the C one? Hand clasping. And then do you have the dominant or recessive of bent thumb? And then when you get to the end here, this is based on how common or uncommon these traits are in the general population. Now, I think when this was surveyed, and this stuff doesn't usually change too much over time, unless the traits are favorable for some reason, and then evolution would make their rates change over time. But I don't think these traits are evolving. These like middle digital hair is not going to increase your ability to survive or reproduce. So these are just basically random traits. If you have phenotype number 1, that is all dominant of all of these that are being assessed here, that's the most common combination of those alleles. And if you have the recessive of each, that's the least common of all these alleles. It's quite uncommon for somebody to have phenotype 32 here, which is all the recessive ones. Most people fit somewhere in the middle, although I've definitely had students that have phenotype. Does anyone here have phenotype number 1? I think we surveyed 0 for one of these, so I don't think anyone can have phenotype number 1. Yeah, no one has a dimpled chin, so, or no, dimpled chin isn't one of these? No, it's not. No one has phenotype 1? No. So I'm phenotype 21. I used to have a list explaining what the actual percentage of the population is for each of these, but I think this is like 2% or something like that of the population has phenotype 21. Anyway, you can see that when you look at these traits and combinations of these traits, everybody, when you look at the traits as a combination, is going to have different various combinations of these things. It's pretty easy once you start accumulating lots and lots of traits for people not to be the same anymore, right? And same thing is true for if you want to use this for genotyping purposes to figure out, you know, whose DNA do I have in front of me. It's pretty easy to go through and say, okay, well, let's assess it for 50 traits. Not too many people are going to have these same exact traits, the same alleles for 50 different alleles, okay? Actually, I think the standard in law is 12. So what they do is they check 12 repeating sequences. Did I mention this previously about how they look at the parts of where your DNA repeats? Okay, so you have sections in your introns, those are the non-functional parts of your DNA, where there's repetitions of things. Those repetitions usually arise during meiosis, during the crossing over events. You get extra repetitions of stuff occasionally. They don't mutate very quickly, so you tend to maintain those through like a family line. But what we can do is look at 12 different locations and say, how many repeats do you have at those 12 different spots? So at this spot on chromosome 1, you have 8 repeats of this sequence. On chromosome 5, you have 12 repeats of this sequence. And then so that combination of repeats at 12 different locations is enough to say, okay, well, that's 1 in 10 trillion or something like that. That's how uncommon it is to have that. And then so for in a court case, if you can say, well, this DNA has these, this DNA has these, so these are a match as far as we're concerned, because it's very unlikely that there are 2 people with the same sequences. In reality, there have been occasions where that is not perfect, because it is possible for 2 people to have the same number of repeats in those 12 loci or locations. They're called loci, locations. But it is extremely, extremely rare. So usually DNA profiling is like the gold standard in law, and you probably already know that. It's kind of cool because when I was a kid, genetic profiling was barely even used in the like legal system. Like people didn't really think it was reliable. And when they did start using it, like juries didn't like believe it. They didn't like understand how the technology worked. So like DNA evidence was not considered to be like good evidence in a trial. Not even that long ago. But now it's the gold standard. People pretty much always trust it. Okay, so this is what we're going to talk about for the next day and a bit. No, normally I would say week and a half, but day and a bit. We're going to look at this idea of genetic variation in the population and the idea of how you pass those genetic variations onto your offspring. So that's heredity. Heredity is passing on of genetic information to your offspring. How do you get what you got? Do you have mid-digital hair? Why? What did your parents have? And what was the chances of your parents giving you mid-digital hair? You could have come out a different way, but why didn't you? Okay, so to reinforce some of the things that we're talking about here, you have 2 copies of each of your genes. Okay, so let's say that this is chromosome 1, all right? Chromosome 1 from mom and chromosome 1 from dad, all right? Maternal and paternal. So you might have at this loci, which is just a place, a location on a gene, or a location on a chromosome rather. That's called a loci. Or sorry, loci is the plural. Locus is the singular, okay? This locus, multiple loci. So at that locus, you might have the dominant allele on one, and you might have the recessive allele on the other. So for example, you might have straight hair on one and curly hair on the other. In that case, you're always going to show the dominant allele. So if this person existed, this would be a straight-haired person. I actually think, by the way, these examples are not taken from anywhere, and I actually think that curly hair is dominant over straight, but I'm not 100% sure off the top of my head. I'm 100% sure I made this up though, so don't take that one as fact. So those locations are one locus or multiple loci. Okay, just places on your chromosomes to where you find this information. Some words that you're definitely going to want to make sure to put in your vocabulary list: dominant and recessive. Okay, allele. Allele is a version of a gene. So one allele would be for curly and one allele would be for straight. Genotype, which is what is the combination of alleles that you have. Is it big B, little b, or big B, or little b? Phenotype, which is what you physically show. And then, very simply here, are they the same or are they different? So if they're the same, for example, if you had big G, oh, definitely shouldn't use red there. If you had big G, that would mean that your two alleles are the same, okay? The same allele on both is called homozygous. Homo just means the same. So if you're homozygous, it means both alleles are the exact same. If you're heterozygous, that just means that the two alleles are different, okay? So this is an example right here of heterozygous. This is heterozygous, big G, little g. Use a different color. Pink, maybe? Oh, yeah. So that right there is heterozygous. So that terminology, dominant, recessive, allele, genotype, phenotype, homozygous, and heterozygous, we're going to use pretty much all the time. You're going to hear these words like 100 times today, probably. So I would call that like a list of core terminology for this unit. Any questions about those, just what those words mean? Okay, like I said, definitely add those ones to your vocabulary list. They're pretty important. Okay, so as I just mentioned here, I'm just going to highlight here what I'm talking about. Chromosomes in a homologous pair code for predetermined character traits, okay? For example, eye color or hair color. Chromosome 14 might have the code for height. Chromosome 9 might have the code for nose shape. Those are not real because both of those things are polygenic. But the reason we don't talk about polygenic traits in grade 11 biology is because to actually figure out what they look like at the end of the day involves some like really complicated statistical math. So we just don't do that in grade 11. If you want to really go into genetics, you want to become a geneticist, doing an analysis of a polygenic trait and what the outcomes will be on that polygenic trait is very complicated. And that's like, you can do your PhD in that. But we're definitely not going to talk about that in this course. What we're going to talk about in this course is we're just going to keep it to Mendelian traits. This gene does this thing, and this gene does this thing, and that's it. In humans, it's not very effective because we don't have many traits like that, but in plants, it works great. Is the berry red or the berry white? That's usually something really simple like that is Mendelian. Is it going to have, like 5 peas in a pod or 3 peas in a pod? That kind of stuff is usually Mendelian. So this type of analysis is very easy to do with a lot of different organisms, just not humans. So I use human examples a lot because people are familiar. When I talk about curly hair, straight hair, people are like, I know what that is. But just keep in mind that for humans and a lot of animals, a lot of this Mendelian analysis doesn't work very well, just as a heads up, okay? Because it's not that simple. Okay, we also talked about the idea here that an allele is a version of a gene, okay? And the place where you find that on the chromosome is a locus. Okay, that's the physical spot where that gene is, chromosome 2 or whatever. Any questions about this stuff so far? I realize I'm repeating myself, but this is, normally we spend a week on this, so. Okay. So this is just an example of a genotype, okay? This is a karyotype. This is like a photo, I guess you could say, of somebody's chromosomes. And you can go through and just say, okay, let's look at chromosome 4. This, by the way, that should say locus, not loci. This locus, okay, is for hair color on 4, all right? This spot. And like, you can look at them specifically. You can even genotype them. So you can, in the lab, you can sequence these. And you can say, okay, the one on the maternal chromosome is big B. The one on the paternal chromosome is little b. So that might be blonde and brown, okay? In real life, that's polygenic, but again, I'm using humans as an example here. Or you could look at chromosome 9, and let's say that that's for eye color, okay? So whereas like brown is dominant over blue recessive, so you look at the person and you're like, okay, both dominant. So if they're showing the dominant trait, if they have both dominant alleles, then we're going to say, well, the phenotype here then is going to be dominant. Okay, dominant always shows over recessive. You could look at the chin structure on 17. Okay, this person has 2 little c's, one from mom and one from dad. If you have both recessive, then you're going to show the recessive trait in your phenotype. So that would be no dimples for this person. Or if you looked at handedness on chromosome 19, you say, okay, well, the maternal is dominant and the paternal is recessive. This person's phenotype is going to show the dominant. So anytime you have the dominant, it's going to show the dominant. So that would mean that this person is going to be right-handed. They're going to show the dominant trait. All of those traits are polygenic in real life. Yes?
Boy, I'm still a tiny bit confused.
Mhm.
Do you know, like how it is like dominant and recessive, or do you just like make it up, or?
How do you know if something's going to be dominant or recessive?
Yeah.
By observation. So you have to have examples of it in the population and look at the 2 alleles. And then you can tell, for example, if somebody in the population has both recessive alleles and they don't show the trait, or they show something, a particular trait, then you're going to assume that's the recessive. Because if somebody's heterozygous, they have both in the population, what do they show? You check. Let's say that they show the dominant trait, or let's say they show blonde hair, okay? They're heterozygous and they show blonde. That would mean that blonde has to be dominant. Anytime there's 2 different alleles and someone shows one of them, that's the dominant trait. More recently, the way we find this stuff out is we genotype. We can actually do this in the lab. We can sequence the genome and figure out what people have, or animals. This is used in farming all the time too, or even in show dogs. People genotype their dog if they're breeding dogs. But not that long ago, 100 years ago, well, even 50 years ago, we couldn't do that. So in order to figure out if something is dominant or recessive, you had to breed things. Essentially, if you take like something that you know is a purebred, that means you know it's homozygous. Both of its alleles are the same. And you take something else. So let me give you an example here, okay? Let's say you took a dog, a black dog, okay? And you know it's a purebred black dog. I'm going to use color here. Let's say capital B is black, all right? So you have a black dog. This is its genotype. And you breed it together with a brown dog that you know is a purebred brown dog. Okay, that means its parents were brown and its parents' parents were brown. You know for sure that it has brown, okay? And you want to know which one is dominant over the other. You're going to breed these 2 dogs together, okay? And when it has babies, they're going to be something, right? In this case, this dog is going to have to give its offspring a big B, right? When it makes its gametes, only a big B can go into its gametes, right? And this dog is going to make its babies with a little b, no matter what, right? It's got to use a little b. It only has a little b to give. So then you look at the baby, which you know for sure is going to have a big B and a little b. It has to have a big B and a little b because it has to get big B from one and little b from the other. And then you just say, well, what color was that dog that you got? If the dog that you got at the end was black, black is dominant. If the dog you got at the end is brown, brown is dominant. Then you'd have to reverse the naming scheme that I just made here. But do you get what I mean?
Yeah.
Okay, so that's how we used to do it for like a couple hundred years. That's how we used to use this information. And this information is important for breeding animals and breeding plants for farming. So you need to know this information when you're breeding plants and animals for farming purposes. Anyway, there's a lot simpler ways to do it now. We can genotype things in the lab, but that's a very recent development. Oh, I didn't realize I actually put this right in the note. I should probably review my notes real quick here before I get started. So here's the definitions for all of these words. I'll give you a second. I'm not going to say it again. I think you guys get it at this point. I'm not going to hit you over the head with a hammer. Did you guys find it hot in here? It's just me? Just me. Okay. I don't want to open the windows because it's pretty cold outside still. It's only 17, but maybe in a little bit we'll open them up. You could have more ventilation in here too. Anyone do anything fun on the weekend?
Nope. Eat, sleep.
That's too bad. I went for an 80K bike ride with my friends, but I didn't make it. I only made it 60K. Then I cramped out. Embarrassing.
That's still a lot though.
Oh well. My wife said it's pretty good. She had to come pick me up though. I didn't make it home.
How's your life going now?
You know what? Not too bad. I'm actually okay. I thought I was going to be totally destroyed today, but it was on Saturday, so I'm actually all right. I can still walk. I'm sore, but definitely could have been worse. It's the first exercise I've had in a while. People are all chained up inside, right? Oh, it's not great. My friends were all very careful. Didn't want to get too close to each other. I guess a bike ride is a good distance activity, but we didn't like ride abreast or anything because we didn't want to get too close to each other. Are we okay, guys? Yeah?
Yeah.
Okay, cool. All right. Let's see, what do I want to use from this? Okay, so what I'm going to talk about right now is the connection between what we were doing previously, which is investigating the biochemistry of this, and which is like the meiotic process. You guys hopefully remember that. Yes, wasn't that long ago. And how that impacts the baby that you get at the end, okay, or offspring. So imagine back here to our to our homologous pair. Okay, so let's say this is chromosome 2. I could pick any chromosome, okay, and you have 2 copies. In interphase 1, what do? I realize normally we spend so much time on this, but like, it is what it is. Does anyone remember? You got to know this, by the way. Yes.
Like the DNA replication.
Awesome. Replication. Okay. So that is what I've just shown here. You'll notice that there's 2 homologs. Okay. They have the same genes on them, which is shown with the same letters. Okay. But they don't have the same alleles because the letters are capitals and small letters. Okay. Some are dominant, some are recessive. But after interphase 1, you have an identical copy of each. Okay, everybody with me on this? So we've got one chromosome 2 and another chromosome 2 plus their identical sister beside them. Okay? Then prophase 1 happens. What happens in prophase 1? This is the super busy one in meiosis where like all the fun stuff happens. Or crazy stuff depending on your outlook, I guess. What happens during prophase 1? So you made a copy, this, and then what'd you do?
Nucleus like gets destroyed.
Okay, so you dissolve the nucleus. That's definitely it. I mean, that definitely happens.
Is there where the crossover?
Exactly. So you get those tetrads forming, right? These 2 are going to hug, and they're going to switch parts between the chromosomes. Okay? So what you're going to end up with after crossing over is something that looks like this. So now you get a mix of stuff. So each of the homologs is not just identical to its sister any longer because you've had some random switching occurring between the homologs. Okay, so now you got like a random assortment of stuff on here. A little bit of your mom's DNA, a little bit of your dad's DNA kind of just mixed together in each of them. Okay, and if you look at these, no 2 chromatid here is the same. Each of the 4 chromatids is just a random collection of traits, okay, that you originally had. So what ends up happening then is that you get these 4 totally different chromatids that you're going to give to your offspring. Where these particular where these alleles go is random. Is this one going to go into the gamete that makes it? Is this one going to go into the gamete? And by makes it, I mean goes on to become a zygote, right? It gets fertilized. Is it going to be this one? Is it going to be this one? Is it going to be this one? Random. Okay, this is a random process. So what that means is that if we look at any particular trait, let's say that we looked at G's, okay? Whatever the G trait here is, this could be literally anything, eye color or whatever you want, okay? If you looked at it, your chance of getting any one of these, big G or little g, is totally random here. You could get this piece, 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 in combination with any other piece on these chromatids, okay? Any combination is possible of things. And your chances of getting any of these is the same. It's totally random which way things are going to go and get into a particular gamete at the end of the day. So because we know that, we can do some statistical analysis to figure out what your chances are of getting a particular trait. So if we know what your genotype is, we can use this information, the fact that it's totally random, to figure out what potential thing your baby can have, okay? So let's say that one of the parents was this parent, okay, this purple parent. In this case, there are two things you could potentially get from this parent. What are the two alleles that you could potentially get? And these are just like letters, yes.
Well, two capital G's, like capital G and a lowercase g, and then two lowercase g's.
Right. So your chances of getting big G are what? What's your chance of getting a big G if you just grab one of these off the table? 50%, right? It's half of them. Half of them are big G, okay? And the same thing for little g. Half is little g. So what you can do is you can use a tool called a Punnett square. We're going to use these a lot, okay? Named after Reginald C. Punnett. So when you see Punnett written, it's always capitalized because it's named after a person. But anyway, this is what you could potentially get from the, let's say that that's the female in the partnership, okay? You could get a big G or you could get a little g, and both are the same chance, right? It's 50% big G or 50% little g. So they're represented in the Punnett square. They each have equal weight in the Punnett square. There's one of each of them because it's 50% one or 50% the other. Now, if you had another partner that also had a big G and a little g, we call this heterozygous. Okay, they have both alleles, big and small, okay, or dominant and recessive. Then on the other side of the Punnett square, you would indicate that, okay? That'd be 50% big G and 50% little g. And then what you do in the Punnett square, let me erase this and I'll go through it step by step. What you do in the Punnett square is you figure out what are the potential combinations of this mom and this dad, okay? So in this column, in the first column, you're only going to get big G's, right? That would be if in this case, mom, I put the mom on the top here, is given big G's, okay? Or in the second column here, this would be if the mom is giving small g's. Now you normally put, if there's going to be two different ones, you normally put the recessive one second. So I'm just going to write it second. Okay, that's just convention. You normally write the recessive second. Okay, and then we can also write here what the father is going to give. So if you're in the first row, the father is going to give big G. Okay, or if you're in the second row, the father is going to give little g. So those are the potential combinations you can get if you put this mom and this dad together, okay? Everybody with me on this so far? Okay, now that we have these genotypes, we can look at what this is going to actually show in the baby. So if you have big G, that means you're going to show the dominant trait, whatever that happens to be. Okay, so let's call that orange, okay? So this is dominant. All right. If you have big G, little g, which trait do you show? What do you show then? Dominant. Okay. So that's this and this. That's going to show dominant. If you have little g, what trait do you show? What do you show then? Recessive. Okay. So in the end, because you had two heterozygous parents, they each had a big G and a little g. If you actually look at the physical trait of the child, 3 out of 4 times they're going to show the dominant trait, and 1 out of 4 times they're going to show the recessive trait. Okay? That's called the phenotypic ratio. 3 to 1, dominant to recessive. Or 75%, that's the same thing. Okay? So this tool, writing it out like this, is called a Punnett square. And we're going to do a number of these Punnett squares together because they help you analyze what you're actually going to see in an offspring if you know the genotypes of the parent. Does anybody have any questions about these before we move on? Just in general, we're going to do a few of these together.
How do you spell that?
Punnett square?
Yeah.
I think I got it written right on here, so. Where are you, Reginald? Really, I didn't write the word Punnett square.
It's in the top. Oh, yeah.
Ah, there it is, yeah. Punnett. Punnett. We'll do some examples of this together. But the important thing to remember is that if the traits you're looking at are on, sorry, what was it? I lost my train of thought. No. Oh, yes. Your chances of getting any one of these alleles is random, right? You could get this one in your baby, you could get this one in your baby, you could get this one, or you could get this one. It's totally random which one makes it into the gamete, right? That is called the law of independent assortment. The law of independent assortment basically means this is a random process. And because it's random, that allows you to do this analysis. Because if this wasn't a random process, if you were more likely to get big G than little g in this process, well then it wouldn't be 3-to-1. It would be like skewed depending on which one was more common. But it is totally random. So because it's random which one you pick, which gamete you get, it's totally random, then that's the independent assortment. Then that means that you can do a statistical analysis and it works in real life. So we use this all the time in genetics. For example, you guys are gonna do a little project on a genetic illness, but that's what we're gonna work on tomorrow. But for example, you can figure out, what are my chances of my kid having this genetic illness? Is it a dominant genetic illness? Is it recessive? What's my genotype? What's my partner's genotype? And then you can figure out, oh, that means that my child will have a 50% chance of getting my genetic illness, or my child will have a 25% chance, or potentially my child has a 0% chance, depending on what your genetics are. Okay? That's how they determine that. And it only works because it's random which of these letters you're going to give to your offspring. So that's called the law of independent assortment. I think it actually comes up later, but I'm pretty sure I asked a question about law of independent assortment on the test, just a heads up. But that's all that it means. It means you can do a statistical analysis because it's random which alleles you get. It's like flipping a coin. So how did we figure this stuff out? Did you guys watch this video, the video on Mendel? No, you guys didn't watch it? Oh, I thought I put that up. Maybe I forgot. Oh no, I did. So you guys didn't watch that heredity video? The one that was in Learning Block 2 from Friday? Okay, if you didn't watch it, watch the video. It's a really good video. And it talks a little bit about Gregor Mendel. Gregor Mendel was a monk who grew a lot of peas. And peas are cool for genetics because just by chance, very lucky for Mendel, most of the traits in pea plants are Mendelian. Well, they're named after him, but they're called Mendelian traits. They're single gene traits. So Mendel was the first person to really figure out how this works, this process. And if you actually go through, I think it explains it a little bit in the video, like how he figured this out. It's really impressive. So he bred pea plants with different traits and kept impeccable notes on what the traits were of the pea plants and what he was breeding them with, like what he was combining. And then he did a bunch of math on the results that he got, the resulting pea plants, and worked backwards to figure this out, which is pretty impressive. So from those observations that he got from raising the pea plants, he figured this idea out of the dominant and a recessive trait. Again, very impressive in my opinion. So what were his original observations that he made from these pea plants? First of all, traits are controlled by pairs of genes. There are 2 for each particular gene. And he figured this out, by the way, way before we even knew what DNA was. Long before we even understood that DNA was what was getting passed on from parent to offspring, which is, again, pretty impressive. So we really had no idea how traits were inherited previous to this. It was basically just like, okay, when 2 people have a kid, it's like a combo of the parents, and just like some of the traits kind of just come together and make the baby, but we really had no idea how it worked. So the second part of this is this idea of dominance. It's called the principle of dominance, which means that usually one allele, one version of a gene, is more dominant than the other and will express over top of another allele. So that's the principle of dominance. That relationship of having a dominant trait and a recessive trait is very, very common in plants. There are some traits in humans that work this way, many, but it's not quite as simple as this always. But in plants, it often is. So Mendel, again, was working with plants, and so this is very important for plant specifically. But humans are a little bit more complicated in their gene expression than this. But in general, there's a dominant and a recessive allele for each trait. So we were just talking about this idea here. So if you have a dominant trait, you're always going to, or a dominant allele, you're always going to show it. If you have the recessive, only recessive alleles, you're going to show the recessive trait, the phenotype. And the last part here is called the law of independent assortment, or the law of segregation. These things are related to each other. They're kind of the same thing. Sure. Law of segregation, law of independent assortment. They're referring to the same thing. So this idea that it is a random choice which of the two you get from either parent. It's totally random because of what happens in anaphase 1 and anaphase 2. Which is, do you remember we talked about independent assortment? So that's going to go one way or the other, or it could switch, and the chromatids could go the other way. They could go this way. So because it's random which way they go, it's totally random in the offspring which one you're going to get. So that's the law of segregation or law of independent assortment, which means that you only get one of the two from the parent's alleles, and it's random which one you get. And again, if you look at the stats, when you do these genetic experiments, that bears out. It's a coin flip which one you're going to get in animals and plants. I'm going to get you guys to come back in a minute and try this question, but not at this exact second. Any questions so far from where we're at here? I feel like I've been repeating myself a lot. Sorry about that. Yeah, normally this is spread out over a week, and we do a bunch of activities in the lab and stuff, most of which I had to cancel. So now I really feel like I'm repeating myself. Okay, so let's do a genetic analysis. Okay? I'm going to use a procedure here to do this genetic analysis, and I would recommend that when you're doing this analysis, you do the exact same procedure. Okay, I'm going to go through it step by step and show you how I'd like you to analyze each of these genetics problems. Normally at the end of this unit, I give you a whole, it's a project on genetic analysis, a whole bunch of different traits. I explain how they're inherited, and I tell you a bunch about parents, and then you tell me a bunch about what their kids are going to be like. Okay, that's sort of like the unit test for this unit. The problem is because of the way we've expedited this unit, I don't think it's a fair test anymore because it's quite complicated. So we're going to skip that and do something a little bit different. But I'm still going to go through the procedure. Okay, so like, how would you approach one of these problems if this like arrived on your doorstep and someone was like, okay, this parent has this and this parent has this. What are the possibilities for their offspring? What could their children potentially look like? Okay, so I'm going to go through the procedure now, step by step, just so that you know what it is. And I recommend that you use the same procedure when you do a genetic analysis. So let's look at eye color. Keep in mind, this is not a true trait in humans because it's polygenic in humans, but I have to make up examples, and it's really hard to find Mendelian examples in humans. So brown eyes are dominant over blue, let's say, for this problem, okay? If you have a homozygous brown-eyed man, and you cross that person, and by cross I mean that they have kids, with a blue-eyed woman, what are the potential genotypic ratios? So genotypic ratio just means what are the potential ratios between the different genes that they could have, okay, or alleles, and what are the potential phenotypic ratios? That is, what will they physically look like? What are the chances of, getting a brown-eyed or a blue-eyed child? That's the phenotypic ratio of the F1 generation. So in genetics, when you're talking about the F1, and this by the way comes from horse breeding. A baby horse is a filly. And so the first generation in a genetics experiment is called the first filial generation. The first generation of horses that you get from breeding, okay? So obviously these are people, but we still call it the F1 or the first filial generation. So the first generation of kids that you get from this meeting of parents is called the first filial, or the F1. That's what F1 stands for in a genetics problem. Okay, so we want to know the F1 generation, the first filial generation of kids. The first thing you got to do is you got to do some let statements, okay? So in my genetics problems, I number these. I number the steps just to make sure that I don't forget any of the important steps, okay? The first step is you have to explain what is your nomenclature. What letters are you going to use to represent these alleles? And really, unless you're given the nomenclature in the problem, and sometimes the textbook does that, it says, oh, this trait, capital H, and this trait, small h, or whatever. Sometimes it does that, but sometimes it doesn't. Sometimes it just lets you create the naming scheme. Either way, you got to tell me what it is. Okay, so I'm just going to create this. I'm going to say, let's let capital B be the allele for brown eyes. Now, when you make that a capital letter, you're implying that brown eyes is the dominant trait in this case. And we know that because it says it right in the problem. Okay, so it says in humans, brown eyes are dominant over blue. So that I know that brown is going to be my dominant trait. So capital B be the allele for brown. Let little b be the allele for blue. Now I've established that's what my nomenclature is going to be, my names. It's like choosing variables in math. This can be x, y, z, whatever, okay? You choose. The next step is to tell me, what do you know about the parents, okay? So we're going to cross, this part in the middle, by the way, you don't need to actually show in bold. So it says P to F1 here. That's parent to F1, parent to first filial. We're going to cross the parent to get the first filial. That's where it comes from, okay? You don't need to write this part. I'm just explaining what I'm showing below. It's a cross of the parents. The parents are being mated together here, which is really weird to say about humans, but that's what this is. You're going to tell me what are the genotypes of the parents. So in the problem, it says a homozygous brown-eyed man. Okay? Homozygous brown-eyed man. Homozygous means both alleles are the same, and brown eyes is capital B. So we know that the man is going to be capital B in this pairing. That's the genotype. Okay? You can indicate that it's male as well, male symbol. Okay? You don't have to. Later on, when we talk, actually we probably won't get to that in this course, but sometimes we discuss traits that are specific to male or female, that is, they're on the Y chromosome or on the X chromosome, in which case you absolutely have to indicate which one is male or female. But in this case, it doesn't actually really matter. So you don't really have to show me which one's male or female, but I can include it. That's the male, okay? And then it says that the female is blue-eyed, right? The only way to be blue-eyed is to have two recessive alleles, right? Because if you had the dominant one, you were going to show brown. So we know that the female in this case is going to be little b, little b. Female symbol. Does that make sense to everybody so far here? All I've shown is what are the genotypes of the parents. This is what they are. Then you need to tell me what are the phenotypes of the parents. Now the order of these actually isn't terribly important. You can show me the phenotypes of the parents and then the genotypes, either way, but you need to include both of these pieces of information. So what do the parents look like? Or how are they expressing their genes? And what is the genotype that they're expressing? If you know it. So sometimes you won't know the genotype of one of the parents. You'll only know that they have the dominant. If you only knew that the father had brown eyes, but you didn't know they were homozygous, then what you'd have to show me is only what you know. Then you'd say, okay, well, I know that one of them's a big B, but the other one could be a big B or it could be a little b. Then you show it like this, okay? That's genotype of father unknown. You know that it has dominant trait, but you don't know whether the second one is big or small. Okay, so that is a potential depending on the question. So you could potentially have that. Okay, so we have the genotypes of the parents and we have the phenotypes of the parents. Now we're going to do our Punnett square and actually cross them. So what the parent actually gives in this case is totally random. Either this parent is going to give a big B or they're going to give their other big B. This one's going to give a little b or it's going to give a little b. I realize that they're the same, but which one they give is random. So you can create a Punnett square. Where's my Punnett square? Right here. So that's step 4. Make your Punnett square. So the male in this case is going to have big B or a big B. That's the same. And the female can give a little b or they can give a little b. And again, I realize they're the same, but it's random which one they get. And then what you're doing in the Punnett square is you're just filling in the combinations of mom and dad. So if mom always gives little b and dad always gives big B, well, all of your offspring are going to be big B, little b, right? It's going to be a combination of the one from mom and the one from dad. I realize this is a little bit hard to annotate on your note. This is probably faster to do from a pen, but I'll try and give you the right amount of time. Are we actually okay with this, or? Yeah. Okay. Is the Punnett square drawn on there? I can't remember. Is the actual like?
It's like just a 4 square.
It's just 4 square. Okay. All right. So next, step 5. What are the genotypic ratios? So in this case, there really isn't a ratio because they're all the same. So this is, in this case, you could just indicate it as a percentage. This is 100% big B, little b. That's the genotype, which is what do the genes look like, or the alleles. From the genotypic ratio, we can figure out the phenotypic ratio, which is what do the offspring actually look like. So if all of the offspring are big B, little b, what are the offspring? What do they look like? What does big B stand for? It's going to show dominant, right? What is it? Brown eyes. Okay? So the phenotypic ratio is 100% brown-eyed. If this union happened in real life, they must produce a brown-eyed child. Anything else is not physically possible, barring some random mutation occurring during meiosis, but that's very rare. But in general, they're always going to display these traits. Okay, so that's the first part. What we've examined here is the F1 generation, the first generation of offspring. Now, in humans, you really only ever talk about the F1. You have 2 parents and you talk about their kids. But in plant and animal breeding, often you breed the children of those, okay? We don't, obviously people are not, their brothers and sisters are not having kids, okay? Or at least not here, they're not. So, but that is called an F1 cross. So in animal breeding or plant breeding, if you take the offspring and you cross them with each other, that's an F1 cross, okay? Now you're taking the F1, the first filial generation, and you're crossing them. So in this case, we're going to cross the first filial, that's the F1, to get the F2, which is the second filial generation. The second filial generation is from taking the first filial generation and crossing them, okay? Again, we don't do this with people, but when you're breeding animals and plants, this is much more common. So the original question only asks about the F1, but I'm just taking this the next step and showing you the F2, okay? The second generation of this. This is, by the way, exactly what Mendel did with peas. He bred the first filial to get the second filial and then used that information to figure out something about the first filial. So every single offspring here is going to be big B, little b, right? Well, the first step 1 is to do what? Before you write the genotypes and the phenotypes. I didn't do it twice here, but what's the first step? What are you supposed to do? What do you need to do?
Let statements.
Let statements, okay? So normally you would do your let statements here, but we just did them for this problem, okay? So I'm not gonna make you rewrite them, but normally you would show your let statements first. Then you gotta give me the genotypes of the parents and the phenotypes of the parents. So the genotypes are big B, little b for both, okay? Because they're both offspring from the same parents and all of them are the same. And they both have the same phenotype because they're identical, so they are all going to be brown-eyed. So you know what the parents look like. Now you look at that genotype and you say, okay, what can this parent give to the offspring? It can give a big B or a little b. So in your Punnett square on one side, you're going to put big B or little b. And then same thing for the other parent. It can give a big B or a little b. So on the other side of your Punnett square, you're going to put a big B or a little b. I'll redo all this, that's fine. Okay, so that would be your Punnett square. Then you fill in your Punnett square. Okay, so this is going to be big B from one, big B from the other. This is big B, little b. This is big B, little b. And this is little b, little b. So when we do our genotypic ratios, we're going to look at what are the ratios of the different genotypes to each other. So in this case, we have 1 big B, 2 big B, little b's, and 1 little b, little b. Okay, so this is a ratio of 1 to 2 to 1. So for genotypic ratios, it's 1 to 2 to 1, big B to big B, little b to little b, little b. That's the ratios of each. Yes, question?
Can you like write it in percentage?
You can write these as percentages after. A number of the genetic problem solving questions ask you to do this and then say, what is the percentage? So in order to get the percentage here, all you need to do is turn this into a fraction. So you want to know the percentage big B, that's 1 over and then the total number of numbers. So there is a total here of 4. So 1 over 4 is 25%, okay? Or if you want to know the percentage of getting big B, little b, it's 2 over 4, that's 50%. So you can express these as a percentage, but generally the problems ask you to express them as a ratio. So when you do this experiment in real life, and Mendel did this experiment in real life, he realized that 50% of the offspring are going to end up being heterozygous, 25% are going to be homozygous dominant, and 25% are going to be homozygous recessive, which is little b, little b. Homozygous members are just the same, okay? So now we can take this genotype and we can figure out what are the phenotypes you get out of this. So big B and big B, little b are both going to be brown, right? So 3 out of 4 are brown. Or you could also say 3 to 1 are brown. That's the phenotypic ratio. And again, this was borne out in a lot of Mendel's experiments. He saw the 3 to 1 ratio of dominant to recessive when you bred 2 heterozygotes together. And then often there's a last step. The last step is just to answer the question. There's often a question saying, well, what percentage is this? Or what percentage of the offspring will be this? Or whatever. So the last part here would be some type of therefore statement. Okay, a statement explaining. So 74% of the offspring are brown, 25% will have blue. Depending on whatever the question is asking you, the question might be asking you something completely different, I don't know, but you answer it in the last step. So whatever that is. There's usually a statement expressing the answer. So our conclusion here is 75% will have brown and 25% will have blue. It's a total dice roll. You have a 4-sided dice. Can you have a 4-sided dice? 1, 2, 3. Yeah, it'd be like a pyramid, but you could do it. 3 out of the 4 numbers on there are going to give you a brown, and 1 out of the 4 numbers is going to give you a blue. Any questions about the terminology here or what's happening? Fairly clear? Okay. One last idea here before I get you to start working on the homework problems, okay? I kind of squished a bunch of stuff together here, which is, it's okay. It just gives you more time to work, because I have a feeling there might be some questions while you guys are doing this. So the last thing I'm going to talk about here is the idea of a test cross. So you asked a great question before, which was, how do you know if something is dominant or recessive? And if someone is showing the dominant, or some organism is showing the dominant trait, how do you know if they have both dominant alleles or dominant and recessive? Okay? The answer is, just by looking at them, you can't tell. You can't tell by just looking at them. But there is a way to figure it out, and this is extremely important if you're animal breeding. Less important now because we can do genetic testing now. But previous to that, if you had a horse and you wanted to figure out if your horse was a purebred for a particular trait, purebred means that both of the alleles are the same. The only way to do that is to do something called a test cross. So we're gonna discuss how you would do that, okay, traditionally. Keeping in mind that this is a very long-winded way of figuring out if something is a heterozygote that you wouldn't do anymore. You would just genotype your horse or whatever. Usually if you're a horse breeder, you probably have money to do that anyway. But so you can perform a test cross. So I'm going to use the idea of these German Shepherds over here, okay? And I'm going to rename them. Rover's not a very good name for a dog. Sorry if anyone has a dog named Rover. I'm going to call this dog Buck. And let's call this guy Betsy. Okay, so we've got a male and a female dog here. We got Buck and Betsy, longtime friends, first-time partners. And we want to know here something about which dog is a purebred. Okay? When we look at Buck and Betsy, they are both black dogs. Okay? And we also know that in German Shepherds, the dominant allele is black. Okay? So they're both showing the dominant. But I want to breed German Shepherds. So I got to check to see if this is a purebred dog. You don't want to use a heterozygote for breeding because you never know what you're going to get in the offspring, which is counterproductive if you're trying to breed purebreds. You always want your purebred offspring to have all of the traits be homozygous for whatever that particular breed is. That's the whole point of pure breeding. So that every time you breed 2 dogs together, you know exactly what it's going to look like. If you breed heterozygotes, then you get a random, combination of things in the offspring, which you don't want. That's the whole point of pure breeding. So not to say that that's good or bad. I don't know if pure breeding dogs, it's not very healthy for dogs in general because they get inbred when you do that. But anyway, it's a thing. So you want to pure breed them. One of these dogs is a purebred. That is, it's homozygous dominant. Okay? And one of them is a hybrid. Now in real life, maybe they're both hybrids or whatever, you don't know. But I know. So I can test to figure out which one is which. In order to test them, what you have to do, and so it's physical inspection, doesn't work, okay? We're gonna look to the F1 generation, okay? We're gonna look at their kids to figure out whether or not they're purebred. So I'm gonna kind of skip ahead here because I'm gonna be repeating myself if I go over that stuff again. So let's start with the let statements. Okay, Capital B, dominant, little b, recessive. Blonde is the recessive trait, okay, for German Shepherds. Now let's look at the genotypes of the parents. So what we're going to do is take our unknown dog, okay? We don't know whether it's a purebred or not. So this is Buck or Betsy, okay? So I'll establish that here. This is Buck / Betsy. We got to breed them separately, not with each other, okay? We're going to breed them with something that we know the genotype of, a blonde dog. If you have a blonde dog, for sure, it's going to be homozygous recessive, because that's what you get when you have two recessive is you get blonde. That's the only way to get blonde dog. Okay, so we do have to know the genotype of one of the dogs, but this one we can tell by looking at the phenotype. So we breed together the unknown dog, Buck or Betsy, we'll do them both, okay, with a blonde dog. If the dog, that is, if Buck or Betsy, that's the dog, was a purebred, okay? That is, it was big B, big B. That's what we want to know, is it purebred? This is what would happen, right? Oh, I have her over there. This is Buck or Betsy. Let's say this is Buck. This is what you would get, right? So here is the blonde dog on the top. This is the partner that we established. And then down here we have the black dog, either Buck or Betsy. So what do you notice about the genotypes of the offspring if the dog is a purebred that you're breeding? What do you see?
They're all going to be dominant traits.
They're all going to show the dominant trait. Every single one will show the dominant trait, okay? So we know that if we do this mating pair and every single offspring dog comes out black, you got yourself a purebred, okay? But let's say the other dog, which is Betsy, is a heterozygote, okay? That is, it has a dominant and the recessive trait. It's still going to look black. When we actually do the pairing, what we end up with is 50% black and 50% blonde. So if you do this pairing and you get any blonde puppies at all, not a purebred. So using this to determine whether or not something is a purebred is called a test cross. You breed it, you breed the animal, and then depending on what you see in the offspring, you decide whether or not it was a purebred. In order to do this, you always have to breed the animal with a homozygous recessive genotype. So it's got to be little b, or little s, or whatever. But you're always breeding it with a homozygous recessive in order to figure this out. Now there's one little complication thrown into this. Let's say that this is Betsy. Okay, Betsy's going to have some puppies with a homozygous recessive dog, just like this. Okay, they're going to do this. And let's say that you get 3 black dogs. What does that tell you? This is a little bit tough. This is a question about probability here that I'm asking. What does it tell you if you get 3 black dogs? Is it possible for Betsy to make 3 black dogs?
Yeah.
Yes. So that is the one thing you got to be careful of here, because this is random, right? This is a 50% black and 50% blonde. So basically what you're saying is, can you flip a coin and get heads 3 times in a row? Absolutely you can, okay? So if you get only black dogs, you don't 100% sure know that it is a purebred. The only way you know for sure is if you get a blonde dog. If you get a blonde dog, it has to be a heterozygote. So the way that this works in reality, is that you have to do it more than once. And if you keep doing it, and you keep getting only black dogs, the chances of it actually being a heterozygote go down and down and down every time you do it. It's like flipping a coin 10 times and saying, can you only get heads 10 times? Yes, but the chances of getting heads 10 times in a row is 0.5 to the 10th power, which is, what is that? I don't know that off the top of my head. Do you guys know? Sorry. Why is my calculator flipping? Come on, flip. Flip. Sorry, one sec. Flip, flip, flip. 0.5 to the 10th power to the 10th is 0.0009. That would be 0.001%. Okay? So that's a very, very low chance of flipping a coin heads 10 times in a row. If there's 10 offspring and they're all black, at that point, you can pretty much assume, okay, we got a purebred on our hands here, okay? But it is true that technically you could flip a coin 100 times and get heads. It is physically possible for that to happen. So you don't know with 100% certainty if you only get black dogs that you're looking at one of these, okay? But the more black dogs you get, the more likely it is that that is a purebred. However, if you get a single blonde dog ever, it's a heterozygote for sure. Because the only way to make a blonde dog is if you have a heterozygote. Okay? So that's the one little hiccup you can throw into this. You can never know for sure you have a purebred, but you can know for sure that you have a heterozygote. Does that make sense? That's a probability thing thrown in there. So just keep that in mind that every single time you make a baby, it's a random selection of one of these four, which means that, of course, you could get the same selection every time, many times in a row. It is physically possible. In truth, when you actually do this in real life, you actually see the stats of it bear out, which is that you get, in this case, you would get approximately 50% of the time you would get one, and then 50% of the time you get the other, because it is random, but it is physically possible. All right. So if you want to know an unknown genotype and it expresses the dominant, you can perform a test cross by breeding it with a homozygous recessive individual. Okay? Then you look at the F1 generation, the first filial generation. If they're all dominant, then you probably have a purebred. Just have to be careful with the way you phrase it. Probably it's a purebred. However, if you see any recessive, for sure it's a heterozygote. Or for sure it has one recessive allele. It has to. Questions about how the test cross works? I'll let you guys write. Sorry. Sometimes I talk too fast. Okay, give me a second. Probably talk too fast a lot. This is the only math in this course, by the way. So if you're if you're not real big into math, this is there's not too much of it. Okay, so I'm going to pause here. I think I forgot to include these questions. Anyway, what you're working on right now, what I'd like you to try is question number 5 on page 193. And at the bottom of page 191, there's a little practice section. You're going to try questions number 1 and number 2. For number 1, use the full 6-step strategy. Remember we talked about first use let statements, then do this. Use that strategy for part A. For part B, you have to work backwards. So there, they're going to give you the result, that is the genotypic ratio or the phenotypic ratio, and you actually have to work backwards up to the parent, okay? So you know what the resultant babies are. Now you need to create your Punnett square and work backward to figure out what the parent would be, okay? So that's for part B. And then, again, for part 2, I suggest using the same 6-step strategy. Just go through from the let statements, okay? That's for the second part here for page 191 on the bottom. Okay, I'm going to give you a while to work on this. I'll give you till 20 after, okay, until the break. Please let me know if you have questions, okay? So if you're going through here and you're like, uh-oh, I'm not sure how to analyze this or I don't know what I'm supposed to get out of this, let me know. Same thing for you guys at home. Just drop something in the chat and let me know, okay? I'll go through it with you where we left off. Did anybody have any questions about the genetics problems from before? So you went through how to look at a test cross. One thing I wanted to point out was if you look at the stuff for today on the website, on Brightspace. There are some little exit quizzes after each section. I updated them this morning, so if you can't see them, I moved some of the stuff around a little bit, so you may just need to reload the content page, but where is it? So there are 2 here. The first one is just some questions about inheritance and vocabulary. Homozygous, heterozygous, dominant, recessive, that kind of stuff. The second part here, this quiz number 2, is more about test crosses and how to do a Punnett square. And after we do this second part here, we're going to talk a little bit about codominance and incomplete dominance. There's another little practice quiz there as well. So the purpose behind these is just to do a little knowledge check. They're not worth marks, but it does help give you an idea of the kinds of questions I might potentially ask on the unit test so that you kind of know where things are headed, okay? So I know that you guys were working on this. Why does it say 4 blank? Okay, let me fix that. That part should say oh, that's the quizzes, right. There we go. Yeah, so that should say these quizzes. Okay, cool. So what we're going to do right now is talk about a slightly different form of inheritance. Previously, I mentioned that there was this idea of, okay, you got your dominant trait, brown fur, and you got your recessive trait, blonde fur. And if the organism has any of its alleles are black, because that's dominant, or brown or whatever, because that's dominant, that's going to be the one that always shows, as long as it shows the dominant. Okay, so a lot of traits operate like that. Okay, so that's the law of dominance, the idea that a dominant trait is going to sort of overshadow a recessive trait. Okay, that type of inheritance is called complete dominance. Okay, complete dominance means that one trait is going to be totally dominant over the other trait, and you won't see the recessive trait. But not every single example works like that in real life. Okay, there are some cases where when you have a dominant and a recessive, they kind of work together and produce something else when you have a dominant and a recessive. Okay, it's like a hybrid of the 2 of them. So there are kind of 2 ways that this can work, and we're going to talk about those. It's 2 totally separate things, but they come out similar. Okay, so the idea here is some of them are incomplete dominance, so where one is not totally dominant over the other, and then the other type is called codominance when the 2 work together. Okay, so let me go through some examples of these, of incomplete and codominance. So like I said, previous to this, we've only been talking about complete dominance, where one allele is totally dominant over the other allele. In examples of incomplete dominance, you get a blend of the two traits when you have two different alleles. So instead of one overshadowing the other completely, you get a little bit of both. So this would be like if you had a black cat and a white cat and you bred them together and you got a gray cat. Okay? A little bit of white and a little bit of black mixes together and it makes a new thing that's like a mix of the other two. Okay? That's a blend. In that case, in incomplete dominance, neither of them is really dominant. Okay, so you don't say one is dominant over the other, because then which one would you choose to be the dominant one? I mean, it's anybody's guess, right? They both are shown in the offspring. So when a hybrid is when you have heterozygous, okay, so two different alleles, shows both traits as a blend, it's called incomplete dominance. Okay, it's a blend of two homozygous traits. Now we'll talk about some examples of this. These are real examples, by the way, actual genetic examples, not made up ones. So in real life, snapdragons, if you've ever seen these before, maybe you want to show you a picture. Pretty. These are snapdragons. Nice flowers. Question?
Could you turn off the lights, please?
Sure. Okay, snapdragons come in a whole bunch of different colors. Okay, we're only going to discuss two of those. So if they have red pigment in them, okay, the allele for red pigment we're going to show as CR. By the way, this nomenclature of using a capital base letter for the trait, in this case it's color, that's why I'm using C, and then using another capital letter to show what the type of trait it is, for example, red. So we've got capital and capital here. This is the proper nomenclature to use for incomplete dominance. Okay, there are a number of questions on this on the quiz, just so you know. Okay, so when you're talking about incomplete dominance, oh, I gotta restart this. This is super slow. These are both uppercase. Shut this off. Did it not save that? No, it didn't. Oh, that's too bad. So anytime you're doing a problem involving incomplete dominance, and you'll know that because I'll tell you, you use, when you're doing your nomenclature, you do a base letter and then a superscript, and they're both capitals. You don't use any small letters, and it's not that single capital and small letter like in complete dominance, because then that's confusing. There is no dominant and recessive, So there is no like one is a capital and one is a small letter. Everything's all caps. So we're going to represent red pigment with capital C superscript capital R. Okay. And white snapdragons we're going to represent with capital C superscript capital W. W for white. So in this case, red snapdragons are homozygous. When you have CR, you get red. Keeping in mind that you always have to have 2 copies. Every organism has 2 copies for every trait. And if you're homozygous for white, CW, you'll be white. But in this case, when you're a heterozygote, one of them is not going to be dominant over the other one because it's incomplete dominance. So what ends up happening when you're both CW is that you get a blend. For snapdragons, that's pink. There are a number of traits like this. And we're going to talk about a type of codominance, which is the second example in humans. But that's like our third topic for today. Come back to that. So the most important word you need to associate in your mind with incomplete dominance is the word blend. If you can remember that incomplete dominance is a blend, you will be far less confused later, okay? It's a blend. It's not both. So it's not some of the petals are red and some of the petals are white. It's a blend. Pink. Okay, the blend is actually a totally new trait, which is a blend of the other 2. Question?
Is this what happened where to a people that's like ambidextrous? Like it's a blend between a left-handed and right-handed?
That's a really interesting question. I don't know the answer to that. I think handedness, though, is polygenic, though it's not Mendelian. If it was a Mendelian trait, then there is a possibility that that is the reason. But I don't think it is Mendelian, though. So it's a little bit more complicated than that. We okay with this so far? That's just one example. Okay. So what's happening here when a snapdragon is CR? Well, one gene is coding for red pigment. In other words, when the ribosomes are going to translate this gene, they're going to build a red protein. And because both copies make red protein, the end product is going to be red. For the CW flowers, both alleles are telling the flower to produce no pigment. If no pigment is produced inside the flower, that's white. Because the petals don't have chloroplasts in them, they're not photosynthetic. So that tissue ends up being white if you don't produce any pigment. But if you get both, you basically get a situation with half as much pigment. And half as much pigment creates pink in the petals. So it's a combination, a blending of no pigment and full pigment of whatever color. I'm sure that blending white with other pigments probably also works. In other words, like if you blended dark blue Snapdragon with a white one, you'd probably get light blue. Because you get the same type of incomplete dominance. So again here, the key is a blend. The third trait is a blend. The heterozygote is a blend. Because this always gets confused with the next one. But if you can remember that it's a blend, then you'll be good. You good? Yeah. Okay. It's a little easy for me to read when people are done writing because they like put their pencils down and stuff, but on the keyboard people just like hold still, I guess. Okay, so that's incomplete dominance. It's when the hybrid, the heterozygote, is a blend. Codominance happens when instead of getting a blend when you mix two alleles together, you get both. So the key word you need to associate with codominance is the word both. Both traits get expressed at the same time. So for example, in the case of short-horned cattle, the hair color gene, which we're going to use the base letter H here, okay, for the allele, for hair color. The red hair color is H superscript little r. Notice that you use, for the superscripts, lowercase letters. That's for codominance. Codominance has lowercase letters for the superscripts. Please be aware that there are many typos in your textbook where they don't do this. So be careful with that. I have contacted the textbook company about that, but there are many cases where they mix these up in the textbook, which is very annoying. But small letter superscript. Okay, so that would be for red hair. And then for white hair in cattle, and by the way, this is real. This is an actual example, okay, of a Mendelian trait in cattle. For white hair, it's H superscript W, okay? And it says recognize this new allelic notation. It's a capital letter base with a small letter, a lowercase superscript. Okay, so watch for the difference here. Just like before, if you're homozygous, you're going to express one of them. Okay, in this case, HRHR gives you a red cow. It's more like brown. To be honest, they're not like bright red or anything, but it's like a brownish red. HWHW gives you a white cow. But if you're the heterozygote, that is, you have one of each, you get both. So both is rowan. Rowan means that the hairs grow in little clusters of red hairs and white hairs. So both hairs actually grow, white hairs and red hairs, in little patches. Okay? It's both. Yes.
So is this kind of like when people have like two different colored skins, they have like different patches? Is that what this is?
Not caused by this.
No.
No. There are examples of these traits in humans. We're going to talk about one of them in a bit, which is blood types. So blood types work like this. Ooh, I'm trying to think of another codominant expression in humans. There aren't a lot. Even when you get people with two different colored eyes, that's usually not due to codominance. There's other things that cause that. I wish there was better human examples. There's just not a ton. But blood types is a really good one. We'll get there. Anyway, codominance, again, the key word here is both. When you get the hybrid, you express both traits at the same time. If this was a flower, it would be some red petals and some white petals or whatever. Okay, but it's always expressing both, not a blend. So in this case, when you have HRHR, you make red because both alleles produce red hair. So that hair is going to come out red, every one of them, because there's only red to potentially use here from the genome, red pigment. And the same thing for white. So in this case, the white is not an absence of red. It's a white pigment. Okay, it's coding for white. In the last example, in incomplete dominance, one of the major differences here is that the white one is actually just coding for nothing. That's what creates the blend. But here, the white is actually a separate pigment. It's a white pigment. And lastly, if you have the heterozygote, some of those skin cells are going to use one gene. The hair follicle cells are going to use the white pigment, and some are going to be transcribing and translating the red ones. So if some are transcribing and translating white, some are transcribing and translating red, what you end up with is some cells doing red and some cells doing white, so you get both. Sometimes you see this in plants that have two different colored flowers on the same plant. So that can exist in plants as well. And it's not only for coloration. Coloration is like a really obvious physical feature, so I'm using that as an example, but all kinds of things could be potentially codominant. It doesn't have to be only color. Color is just an obvious one. So I'm going to go through a little example here below of what it looks like if you cross two roan cows. Roan, remember, is the heterozygote. One of the alleles is red and one of them is white. Okay, so I'm starting with my let statements here. Capital C, small r is going to be for red fur, and capital C, small w represents white. Oh, sorry, I'm crossing a red and a white cow here. I think I actually crossed the first filial generation too. Yeah, I'm gonna cross red ones afterwards. Those are their kids. So first of all, let's say that I crossed a white cow and a red cow. Sorry, my notes didn't print in color. That looks black, but it's supposed to, it was red. So just like before, you're gonna create your Punnett square and put the alleles for one on the left and the alleles for the other on the top. Okay? So here I've got my red cow on the top and my white cow on the left. What do you notice about all of the offspring of red and white cows? What do you notice?
They're the same.
They're all the same. Okay, they're all going to come out with the same genotype, and therefore they're all going to come out with the same phenotype, which is roan. Okay, so all of these cows are going to be roan. That's the F1 generation. That's these guys right here. There's my attempt at red and white fur. So if I cross my F1 generation, this is to get the F2 generation, now I'm going to take a roan cow, CWCR, and a roan cow, CWCR. I'm going to cross them. Okay, so if I mate these two together, I'm going to get this. And we can look at our genotypic and phenotypic ratios here. So, CWCW, that's going to be what? What'll that be?
White.
White. No surprise there. The white highlight doesn't really do much. I guess that makes sense. What can I use instead? Gray? Sure. Let's call that white. Okay. And CWCR. What's that going to end up being? Roan. Okay. Mix. And CRCR. What's that going to end up being? Go ahead. What's that?
Red.
Red. So you'll notice that the actual outcomes that you get look a little bit different than they do in the complete dominance examples, which we did previously. So instead of getting a 3-to-1 ratio when you mix the heterozygotes, here we get a 1 to 2-to-1 ratio when we mix the heterozygotes. Okay, because the heterozygotes look different than the homozygotes. So we end up with this, which is a 1 to 2-to-1 of white to roan to red. Oh, it's asking me to predict it on the side here. Whoops. Well, I jumped ahead. I just did the whole problem. No problem. Oh, the Lancy. Any questions about how this works? So these are two new types of inheritance. We started off talking about complete dominance, where one allele is totally dominant over the other. In incomplete dominance, when you get a mix, the mix is a blend. And in codominance, when you get a mix, you get both. Okay, expressing at the same time. 3 different types of inheritance: complete dominance, incomplete dominance, and codominance. What I'm going to get you guys to do now is try some examples here with incomplete and complete dominance. It's going to get you some of these problems you work from the top down, where they give you the parents and you figure out the offspring. And in some of the examples, they give you the offspring and ask you to work backwards and try and figure out what the parents would be. Okay, some of these are a little bit complicated. If you're having trouble with any of them, just let me know, okay? I can do some examples with you too if you like. So try these. On the online course, is this gonna work? consider watching this video, which is a summary of incomplete and complete dominance, okay? There's lots of good diagrams in here where they explain the process in a little bit more detail, okay? I highly recommend it. Although I suppose you don't have to watch it, but I do recommend it, though. It's a good video. There are the practice questions, and then there is a little practice quiz that you can do. I recommend doing that as well, because it's going to give you some problems to say, what percentage of the kids would be this in this example? It's like you should be able to solve that. And if you can't, that's an indication to you that you should go back and take a second look. Before you start this, I got one more thing for you to do today, okay? There is a third lesson, and I had to squeeze this one in because we're running out of time for this unit. So this is a pre-recorded lesson, okay? I don't think it's super long. I think it's like 25 minutes. I actually pre-recorded this one last semester. So it mentions a couple things in the video about, for example, I talk about how if you're falling behind, like, don't worry about it. You don't have to do this content, et cetera, et cetera. That was for last semester, okay? So you do have to do it now, unfortunately. So we're not in the same boat. You guys know how it worked last year, right? Yeah. So this is a little bit different now. So just be aware of that. I did record this last semester, so don't be confused. But I mean, the content's exactly the same, okay? So what I'm going to be discussing here is the idea of blood types and what happens when you combine types of inheritance together. So sometimes there will be 3 alleles for a particular trait instead of only 2. And 2 of them might be codominant, and then the other one is dominant or recessive to the other 2. Okay, so now we're actually starting to combine more than one of these types of inheritance together into a single example. And blood types are a really good example of that. So before you watch that lesson video, watch this quick little summary. I think it's about 8 minutes long, and it explains how blood typing works, okay? What are the blood types and how do you get the various blood types genetically? Okay, so start with that. It's a really good TED Talk that talks about blood types. And then I'm going to go through the note on how to actually use that to problem solve and figure out what a person's blood type will be, okay? So we can do genetic problem solving just like we can with any of these other examples, but just do it with blood types. But there is just a little bit of extra there that you need to know before you do that type of problem solving. Okay, so there's a lesson right there, and then there are some practice questions that go along with that. I realize that that's more content than we normally do in a day. I'm a touch stuck here because we had to finish this unit tomorrow. So do your best. Do your best to get through that. We might have a little bit of time to finish this in the morning. To give you an idea of what we're going to work on tomorrow, our first task is I'm going to take up some of these practice problems, some of these blood type practice problems. They're the ones on, let me throw it up on the notes. Okay, so here's the blood typing stuff. These practice problems here, I actually take up in the video. So you pause the video, you try these practice problems, and then I take them up in the video, okay? I highly recommend that you go through the take up together for these. Plus you can fast forward around and stuff because that can be long-winded, okay? Feel free. And then there are these practice problems at the end here, 2 through 4, or 2 through 5? 2 through 5, okay? Try them. Those are the, I think those are the ones that are mentioned here. Did I mention it on the thing too? 2 through 5? Should have. Oh, for God's sake. No, I should put that in there too. Okay, so then there's practice problems 2 through 5 that are in the actual workbook. Okay, I'll add that on there. Try them. I'm going to take them up at the beginning of class tomorrow. I'll work through the problems with you, okay? Then tomorrow morning, we're going to do a sort of like an independent reading lesson where you guys are going to read about some genetic disorders. And in the second half of tomorrow, you're going to work on your project for this unit, which is you are going to design a tool to explain meiosis to another student, okay? So how you do that is we'll leave it a little bit open. You can make a little video. You could potentially, I guess, make a poster or something like that digitally. Well, I'll leave it open for you. Some people last year made a song. You can, I guess, technically do a song. It doesn't work very well, but absolutely you can do that. We'll go over and explain how it's going to work. But you are going to design like an educational tool to teach somebody else about the process of meiosis, okay? How to make a gamete. And we'll talk about that in more detail tomorrow. Okay, any questions about what you need to work on today? I realize that some of this problem solving stuff is a little bit tricky. So if you're stuck, don't be afraid to say something. That goes for people at home too, okay? So just let me know. I can jump into a little video conference or something and we can go over a problem together. But I am gonna take up these problems right here. Where are they? Right here. These problems with you tomorrow, okay? We'll all take those up together. Okay, good luck. Let me know if you need anything. Quickly, guys, I'm just going to run through question 5 from the homework because like a number of people are asking me online as well about number 5. So we're all just going to do it together real quick here. So number 5 is asking you about thalassemia. Okay, there's 3 potential states for thalassemia. There's major thalassemia, which means you are experiencing a lot of anemia. It says it's anemia, right? Which is a lack of ability to transport oxygen. Okay, so you've got major thalassemia. Oh boy, I'm gonna misspell this. Is it 2 L's? What's that? It's not showing up there? Why not? It's on mine. Okay, now it's on a huge delay. No? Not gonna do it, eh? One sec, guys, sorry. There we go. Okay. You got major thalassemia, you got minor. Thalassemia. I'm gonna get super lazy and copy and paste that. And then you've got neither, which we're just gonna call healthy, okay? So that just means you have no thalassemia health condition. So the first part of the question is asking you which type of gene expression is this? Okay, which type of inheritance? Is this expressed as a dominant recessive relationship, which is called complete dominance? So complete dominance, if you remember previously, was when you could express it with 2 alleles, a capital letter and a small letter. One is dominant to the other, okay? Or is it incomplete dominance? Or is it codominance? Okay? In complete dominance, you've got A. The 3 genotypes that you can have are AA, AA, and AA. But in this case, in complete dominance, you've got this is dominant, and this is recessive, right? So there's only 2 potential phenotypes that you can get here. But in this problem, there's 3 phenotypes. These are the 3 phenotypes, the 3 things that you can be. So it can't be complete dominance. You can't get 3 phenotypes from that. Incomplete dominance. Incomplete dominance would be like if you had, okay, anemia healthy or anemia, let's actually write the full genotypes out here. Anemia healthy, anemia thalassemia, or you could potentially have the hybrid. And if you have the hybrid, because it's incomplete dominance, you'd get a blend, right? This is the blend. So what would a blend look like for thalassemia and healthy? And I mean, like, obviously this is a hypothetical here, but if you're looking at this here, I think minor thalassemia would probably be a good way to describe a blend between healthy and major thalassemia, right? Major anemia or no anemia, a blend between the two would be minor anemia, right? That's a blend. So this is our front runner here. This makes a lot of sense. Okay, that's incomplete dominance. Or it could potentially be codominance. Codominance, remember, is both. So you could have healthy, now I'm using lowercase, by the way, superscripts here, or you could have thalassemia, or your hybrid in this case would be both. So the question is, what would both look like? Can you be thalassemic and healthy? Can you be both of those simultaneously? For codominance, you have to be both at the same time. So in this case, I don't think that's physically possible. Can you be both healthy and thalassemic? No, both of those can't be simultaneously expressed. So because both of them can't be expressed at the same time, this is not codominance. So the only thing it can be in that case is incomplete dominance. The second part of the question is asking what are the genotypes that are involved, which is exactly what we just wrote out here. Does that make sense, Connor? Any questions about that? The type of expression? Okay, cool. That was a good example to do together.